Thank you very much. I'm glad to be here, and I want to share with you some information about our practices at the JBC, particularly regarding Western blots. So I'll talk about that and our experience at the JBC and what our expectations are for Western blot. But first I would like to talk about the issue of reproducibility in general. And many of you are aware of some of the problems. There have been concerns about whether published research findings are false and is science really reproducible. It's been estimated that a large fraction of preclinical research presented in high-profile journals is irreproducible. There's a need for all the major stakeholders in the scientific enterprise to address these concerns because we don't want to risk lack of funding and public trust. We need to support our scientific enterprise. All of the stakeholders should ensure compliance with good scientific methods and guidelines for data presentation, and researchers, like all of us, should be trained in experimental methods and design, data selection, data analysis, inclusion of controls, statistical interpretation, reagent validation, and experimental replicates and repeats. So that's what I'm going to teach you a little bit about today, I hope. So these are some results of a nature survey, and of the people who were polled, they were asked, is there a reproducibility crisis? And more than 50% of researchers said yes. And another question was, have you failed to reproduce an experiment? And in chemistry and biology, we see a large fraction, particularly with other people's experiments and even sometimes in the same laboratory. This is also true in medicine and other areas as well. So the National Institutes of Health in the United States, which is the major supporter of biomedical research, is doing a number of things. There was a joint workshop with Nature Publishing Group and Science, and they endorsed a set of principles to enhance rigor and reproducibility. You can look that up if you'd like to. It's bit, ly, and so on there. And there's also been a response by the United States National Academy of Sciences. There's been a consensus report entitled Fostering Integrity in Research, and so if you have detrimental research practices, these may not be considered misconduct, but they damage the research enterprise. So what are these detrimental research practices? They deal with authorship, statistical analysis, not retaining or making data available later, and also neglectful or exploitative supervision in research. We'll talk mainly today about data and how we handle it. What are we doing at the JBC? Of course, the journals are some of the major stakeholders in the scientific enterprise. We are trying to put out the most reliable information we can and publish very accurate information. This is an article that came out a couple of years ago, and you can find this on the JBC website in 2015. Transparency is the key to quality, and this was written by two of our associate editors, Dr. Fosang and Dr. Colbert. So they talk about a number of things, and we have revised our guidelines at the JBC to improve rigor and transparencies. There are three major areas. One is disclosure of experimental design and reporting of experimental uncertainty and reproducibility. So, for instance, when you talk about 3 experiments, it doesn't mean just taking the same sample and putting it into the instrument 3 times. That's going to be very close, but we really want you to say 3 biological replicates, that is, doing the experiment 3 times. We've also changed our guidelines about improved statistical and graphical representation of quantitative data. We would like to have people show the individual points and use error analysis in a different way. We don't like standard error of the mean. We prefer standard deviation or something like that, and using plots like this where you actually show the data. And then I'm going to talk about today is that we've revised our guidelines for the presentation and quantitation of gels, particularly immunoblots. You can see a lot of deficiencies with some of these, and I'll talk to you primarily about this today since this is really mainly about Western blotting. So let's talk about our experimental procedures then. And the first thing is we really need people to provide adequate information, and particularly the antibodies that you use, because the antibodies, the quality of the antibodies really determines everything. The description should define the species of animal you're using and the source of the antibodies, including even the catalog numbers and lot numbers, because these can change a lot and that may have a big effect on your specificity and your results. And you can consider using research resource identifiers to unambiguously identify the antibodies in your study. So there's a website here, scicrunch.org, to use this procedure. It's also important to provide the dilution information. This will help readers repeat the experiment. So put yourself in the place of a student in another lab and trying to repeat this work. So the more information you can provide, the better, easier it will be. So this is a good way of doing this. This could be done, for instance, in the supporting information to your paper, indicate all the antibodies, where they came from, what the dilution was, and the vendor, and even the catalog number would be good. You're describing a novel antibody. Well, this means an antibody that you made, or you had it made for you. And is it a monoclonal or a polyclonal? And what was the antigen used? Was it a peptide, or was it the intact protein? And if it was a peptide, what's the region from the protein, and how was this conjugated then? Also, what animal species was the antibody raised in? You know, was it mouse, rabbit, sheep, goats, chickens, whatever? And if you fractionated this, what Ig classes are ready? So this is actually very important information as well. Also, describe if the antibody was purified and how. How do you purify antibodies? Well, there are several ways. The 2 main ways, one is with protein A, and that will, of course, get all of the antibodies that you're interested in then, so you're not separating just one IgG, but many. The other way is to use antigen-antibody purification. We've done this in my own laboratory, where you actually take your antigen and couple it to beads and then bind all the antibodies, then elute at low pH so you get a specific antibody. But you have to describe how you do these, because it's very important for other people to repeat this, particularly if it's an antibody that you made in your laboratory. It's also important to demonstrate the specificity of your antibody. So this is how you can do it. And are you describing a novel antibody, one that you made? So you can do this in a normal reaction. There can be false positives here. So for instance, this isn't a real interaction. You will get a response, but it's not really true. This is very important. You can use genetic strategies. In principle, you could use a knockout some other ways, peptide blocking, and also mass spectrometry, for instance. I'll talk about that, or you could do protein and peptide arrays. But whatever you do, you need to describe it and show that your antibody is specific. Okay, so what's evidence of specificity? This is from one of my own papers, and these are Western blots, and this is an old paper. It's from 1982, so this is 36 years old. How many of you are older than 36? No, just a few. Okay, so most of you weren't even born when we were doing this. In fact, at this time in this paper, we actually did, the term Western hadn't been used. They were just immunoblots. Actually, I've never liked the term Western anyway. That came later. But what we did, we used, these were microsomal samples, and we made our antibodies, and you can see that we're only picking up one band here under these conditions. So these are actually highly specific. You don't see too many gels like this today. In fact, I think when we started, we thought everybody would be doing this to show that they had specific antibodies, but it turned out to be much easier just to cut out a zone and show these. But these show you that we're really dealing with highly specific antibodies. Also, I'm pretty sure this was the first paper in which anybody tried to quantitate the response, as far as I know. At that time, we actually borrowed another colleague's densitometer for thin layer chromatography plates, and it seemed to give a good response. And here are the peaks, and so this is a linear range, and this is how we got started doing quantitative work. I'm going to come back to this later, because even after 36 years, it's very important to be working in this range and not out here, because you can saturate your response if you put too much sample on. Here's a more recent demonstration of specificity. This is also from my laboratory, and it's just from last year in JBC. And this is a protein we used as an antigen. It's a cytochrome P450 enzyme. This enzyme is in human skin. And as you can see, most of the different skin samples are giving two bands. So how do we know that this is really specific in this case? Well, we actually cut these zones from the skin samples and submitted these to proteomics, and we found the protein in both cases. We actually got very extensive coverage. So we're pretty sure, we actually think that the low band is the mitochondrial form, and this is the cytosolic form, and there's a matter of processing. So we're still working on that. We couldn't find any response in liver or kidney, so we think it's specific for skin. So again, we don't have a requirement for showing full gels yet at JBC. I know some journals actually do this, and it actually wouldn't be a bad idea, because if you have antibodies this clean, you can really show people that you're working with good antibodies. Okay, well, let's talk about JBC and what we expect there. So the figure presentation, this is very important. All you have to do is go to the JBC website, and we have, I think it's a pretty easy way to follow it now. So you actually go there and you'll find a section called Collecting and Presenting Data. I think it's under Instructions for Authors. Now what's important here, whoops, let me go back, is the section Collecting and Presenting Data. There'll be a number of sections here with all of the specific requirements that we have at the JBC. So here we have some different blots, and these are, as you might guess, good and bad examples here. So which set of blots is more informative? This set or this set? How many people say this set? Well, you're right. These are obviously better, Here I really can't tell what's going on here. They don't even, the whole gel isn't even shown, and we certainly don't know if this is a very specific response. It's a little better here. One thing that's also very important, we want you to show a molecular weight marker above and below the bands of interest. So we actually really want you to have two markers in all of your gels so that other people will know what zone we're really dealing with this. But don't show things like this, because we actually now in the front office at JBC, before a paper is finally accepted, your images will be reviewed, and if you have something like this, you will probably be asked to send new figures, or perhaps even your original data. So it's best to actually save your original data and work it up properly to start with. It can be sometimes in color, it's useful, sometimes it's not necessary. We don't charge for color prints in the JBC, so color is fine. Okay, how should these be presented? Well, again, I already made this point, but there should be sufficient area around the bands, ideally at least one marker above and one below the band of interest, and the immunoblot should contain the positions of the molecular weight markers. If you're using two gels, then show also the isoelectric point ranges, and the images should be appropriately adjusted such that you retain sufficient background information. I think, okay, what do I mean by that? Well, basically what you can do here is lower the sensitivity by changing the histogram and get rid of all the background, but then you'll probably be getting rid of some of these parts here that may be important, and of more concern, you may be eliminating some bands that are important for your reader to see then. So don't overly contrast your gels before you make your final publications. Here's another problem we see a lot of, and that is with splicing. What do I mean by splicing? Well, here we have the original data. For some reason, you run these, and we're really not interested in the one in the middle, the set in the middle, so we only want to show these and these. So if we take this, then we can show it like this. We put this together and this, but put a line here. Don't show it like this, because this is not acceptable. This will imply that these were run at the same time, which isn't really true. So put a break in the middle of the gel if you're going to put these things together, and also indicate that in the legend as well. Because if we see catch you doing something like this, we will return your figures to you before your paper can be accepted. So you might as well do it right the first time. So again, this is the original data, and again, you should never splice between two different blots, and also results from two different blots are not directly comparable because there are different transfer efficiencies, blotting conditions, etc. So this is not acceptable, and in fact, I don't know if those of you in the front can see, this is actually obvious to my eyes that this was spliced together. And actually when they check this at the journal, they put this through the computer, and you can't really fool these people, because the software will definitely show you that there's been a problem. Okay, so here's another important thing, and we're trying to get people used to this too. So one of the problems is loading controls. One of the problems is you see many examples where people are using actin or GAP-DH or something as their loading control. We want you to quit doing that. Why? Because, for instance, with actin and tubulin, your cell conditions may change, and here you can see that under these conditions, the actin band has been reduced by 20%. So it's not really a very good standard. And the same thing for tubulin, or probably GAP-DH too. So what we really want you to do is use total protein. And here are, this is an example here. This is a list of studies demonstrating the variation in loading control expression. So you can see here that with actin, tubulin, and some kind of voltage-dependent anion channel, all of these things can change between experiments under different conditions in cells, and so they're not really very good standards. So there are good ways now of actually doing a total protein stain. There are a couple of different fluorescent stains you can use, and then using the Lycort or other equipment, basically just normalize everything to the total fluorescence for total protein. So this is, they're usually not linear here. So here's an example. This is some loading controls, and here's the problem, because here's tubulin, and also it's the problem I mentioned from way back in 1982 that I told you about. We already recognized it then. You have to work in this range. If you're up here and you're saturating here, this value can change depending on the system, the detection method, and you're out of the range. So you actually would like to be in this range then, but what's really good here, this is a total protein stain, and this is typical data for different systems. This is actually much more linear. There's a much broader linear range, and if you're doing total protein, this is less likely, this is not going to really change between the different experiments you're doing. And it's actually much easier to do this, frankly, because you can actually strip and do your blotting and do this, and it won't interfere with your antibody stain. So do it this way. We're trying to get people used to doing this, and it's really in your benefit to do that. Okay, this is something I already touched on. One of the problems is signal is not reflective of sample concentration outside of linear range. So, for instance, again, here's the issue. Here's a bunch of blots, and then it turns out if you look at the histogram here, this isn't really balanced very well. This is way too dark to actually do any kind of quantitation. I think this is actually, yeah, this is actually the same gel. So these basically all look the same, whereas over here, this is what they really look like when you've actually normalized it so that you're using your pixels correctly. Then. So now we actually have a threefold difference, and we're actually seeing a lower band as well. None of this is actually visible. So make sure that you are in the linear range of detection of your protein and also the total protein as well. Another issue we try to deal with, and that's post-translational modification antibodies. So basically, as I'm sure you all know, there are many ways of modifying a protein now. I think I know there are over 100 different ways, all kinds of things you can do to attach things, and there are antibodies that recognize this kind of modification. So if you're doing that, these should be normalized to the total unmodified protein, not to actin. You should actually, for instance, in this case, STAT1 is the protein. Here's the phosphorylated version. This is actually not in the linear range, I don't think, but we do have the markers here. So basically, you normalize the amount of phosphorylation based on the total stat here. You can see that this would be deceptive because whatever conditions these are, basically you can see that this is higher under these conditions, so is the p-stat. If you use actin, basically you'd be normalizing to this, but you really should be normalizing to this, okay? And so remember that crop your blots appropriately, don't cut them too close, indicate all splices, normalize to total protein loaded, and then if you're using post-translational modification specific antibody, please normalize these to the total unmodified protein. So these are simple rules to follow. They're really not hard, but if you do these things this way, you will save yourself a lot of time and trouble when you go to publish your paper, and you will have a better paper. And oh, very important also, this is something I have to emphasize over and over again to the people in my own laboratory: save your data and save multiple copies of it. Now save your unmodified, uncropped data. Why? Because you may actually have to give it to the people at the journal if there's actually a problem. You want to save it anyway. So if you have film, you scan the entire blot and save it as what's called a TIFF file, but also retain the film. With imaging systems, you export a copy as a TIFF, also retain a copy of the image system in the native format, whatever it is, and TIFF is preferred over JPEG. I won't go into that today, but it's actually much better and much higher quality. But never, never archive data in PowerPoint. PowerPoint is too low a resolution, and basically the only thing PowerPoint is good for is presenting presentations, not for handling your data, and not for making figures. So never use PowerPoint for this. Use something like TIFF or at least JPEG, but we prefer TIFF. And also save your original files as they come out of the machine. So this is the last slide, and basically I'll try to answer any questions you might have. I'm going to leave this up. Remember that we have these resources here you can find at the JBC, and also this is the magazine that comes out every week from the American Society of Biochemistry and Molecular Biology, and you can, Dr. Keira Sakabi, who is in charge of our image analysis at the JBC, has written a number of articles about this, and again, in the author instructions, they're also here. So I'm going to leave this slide, I think that's the last one, and I'll try to answer any questions you might have then, but I'll leave this slide up if you want to copy this down. Thank you. Shishi.